Welcome back. This is day 6 of genetics, and today we're going to be figuring out how do mutations arise in an organism. Now, we've actually seen this word mutation before, right? And we said that a mutation is basically just a change in the DNA sequence. So today we're going to be looking at a few different types of mutations, as well as how they come about and what effect they have on amino acid sequences. But before we get to that, you guys have got 4 do now questions. So go ahead and pause this video, answer questions 1 through 4, and press play when you're ready to review them. So number 1, describe the main function of this process. Well, in order to describe the function, you had to figure out what even is this process. So looking at this image, we see that we're starting out with one DNA molecule. It's double-stranded, it's DNA. But then that DNA actually unzips and is used to create 2 identical double-stranded copies of DNA. So 2 identical copies of DNA. So what is that process called? You could say maybe we're duplicating or replicating DNA even. So hopefully this is ringing a bell. This process is known as DNA replication. And DNA replication has to occur in order for mitosis to be successful, right? We know that mitosis, we're starting out with 1 cell and we're ending with 2 identical cells. But in order for those cells to be identical, they have to have an identical copy of DNA, right? So DNA replication must occur for mitosis, which is used for things like cell growth, repair, and replacement. All right, number 2, what would be different if the process pictured were transcription? So transcription is the very first step of protein synthesis, right? And we know that we're taking a copy of DNA and turning into a single-stranded copy of mRNA. And there lies the difference. If this were transcription, we would see us ending with a single-stranded copy of mRNA instead of this double-stranded copy of DNA. All right, so double-stranded copy is DNA, single-stranded is mRNA. All right, number 3, transcribe the DNA sequence, right? So using this one, let me just try that one more time. Because this is transcription, we're taking that DNA sequence and turning it into an mRNA copy. So using our base pairing rules, you should have gotten that mRNA sequence is UAU, GGU, and GAA. All right, number 4, use the genetic code chart below to translate the RNA strand from number 3. I'm actually going to go to the next slide. You guys should have this chart on your paper, right? Remember, this is what we use to figure out the amino acid sequences using our mRNA codons. So I wrote down our mRNA codon right here. Now, we're going to use those codons to figure out what amino acids are coded for. So I'm going to color code each of our codons. Let's do the first one as red, second one as blue, and third one as green. So you may notice that this codon chart is broken into 3 sections. You've got the first base on the far hand on the left side, the second base on the top, and the third base on the right. This is the order in which you're going to look for that amino acid. The first place you're going to look is the left-hand side, then you'll move to the top, and then you'll move to the right. So I'll show you what I mean. The first codon we're looking for is UAU. We've used this codon chart before, so I'm going to move a little bit faster than I normally would, but I see that my first base is U. My second base is A, which means I'm going to be looking in this box right here. And I see that UAU actually codes for TYR. Right, our second base, or our second codon is GGU. So again, first base G, second base G, which means I'm looking in this box right here. And my last base is U, which means GGU codes for GLY. All right, and third base, GAA, I'm going to go to the left and find oh, I'm sorry, I made a mistake. GAA, there we go. I was like, that doesn't look right. Perfect. So GAA, we then see it codes for GLU. All right, so the amino acid sequence you should have gotten is TYR, GLY, GLU. Right. So the very first thing we need to do today, well, I guess it's now the second thing after your do now, is run over some vocabulary words we need to know about mutations. So I'm sure you'll be shocked to find out that the very first word I want you guys to write down is mutation. So mutation is a change in the sequence of DNA that is due to an error in replication, environmental factors such as certain chemicals or radiation, or even intentionally biologically engineered. So sometimes mutations happen just because our DNA polymerase makes a mistake, right? Just an error in replication. Sometimes we are exposed to environmental factors that can cause mutations. That's why we have to put on sunscreen, right? So we don't get mutations brought about by radiation. And sometimes we can even intentionally cause mutations in DNA sequences. So in just a few days, we're going to be talking about genetic engineering, where we as scientists can actually intentionally manipulate DNA to cause mutations, right? So there are several different types of mutations that we need to know, but thankfully they're named according to just some common sense. So these vocabulary words won't be too difficult for you guys to remember. And I'll show you what I mean. So when a base is lost or deleted, we're going to call that, you guessed it, a deletion mutation. Okay, when a base is inserted, we're going to call that an insertion mutation. See, thankfully, these words are pretty easy to remember. When a base is lost, it's called a deletion. When a base is added, it's called an insertion. Now, deletion and insertion mutations can actually cause what's called a frame shift, meaning that that reading frame shifts or changes, which actually changes the entire amino acid sequence. And I'll show you a picture of what that looks like. So here at the bottom, this top strand is our original DNA strand. So I'm going to break that down into codons, right? Series of 3s. And remember, these codons are what code for amino acids. But then if there's a deletion, if this T base right here is deleted, let's see what happens to the rest of our codons. So our first codon stays the same. We still have TAA, but then look what happens here. We see that although this first codon is the same, these next two codons are actually different. So CTG is not the same as CGC, and CAG is not the same thing as AGG. So because we just lost this single base, it actually caused the entire DNA sequence to shift, which is going to change the amino acid sequence. So deletion and insertion mutations can cause a frame shift, which is going to cause some pretty detrimental effects, right? It's going to change the amino acids that are coded for, which is going to change the protein that's coded for, right? And the last mutation we need to know is when a single base is substituted for another. I know you'll be shocked to find out that that is called a substitution mutation. So thankfully, deletion, insertion, and substitution are pretty straightforward when it comes to mutations. But there is one more word we need to know, and I think I've actually brought this up in a few of my classes. Now, if a substitution does not change the amino acid, this is what we call a silent mutation. So remember that there are multiple codons that code for the same amino acid. Right. And we can see that when we look at our codon chart. And in fact, in one of your notes before spring break, we said that this is actually serving as mutation insurance for your DNA. So if there is a mistake, if there is an error and one base is accidentally substituted for another, sometimes it doesn't always cause an effect. So we call this a silent mutation because we don't actually notice it's there, right? If someone is silently sitting in your room, you may not notice that they're there, which is kind of a scary thought. I hope there's no one in here. But the same thing happens with your amino acid sequences. If there's an accidental substitution that doesn't actually change the amino acid, you're good because the same protein is coded for, which doesn't affect you, right? So silent mutations, we may not notice they're there. And the reason we may not notice they're there is because multiple codons sometimes code for the same amino acid. And that same amino acid means the same protein. All right. So now we're going to do some examples with mutation practice. So the very first thing we're going to do, I've given you an original DNA sequence. With this original DNA sequence, we're going to find the mRNA and the amino acid sequences. And once we've established that, we're then going to compare it to our mutated DNA sequences. And we're going to figure out, will there be effects based on this mutation, as well as what type of mutation is this? So let's go through the first example. So the first thing I want you to do is actually pause this video and tell me, what is the mRNA sequence of this original strand going to be? Press play when you're ready to review it. So that original mRNA sequence you should have gotten is AUG, UGG, AAC, CGC, UGC, and UGA. Right? Now, same thing, pause this video and figure out what amino acids does that sequence code for. Right, now, I'm not actually going to show you how I came to these answers because hopefully by now you guys know. And if you don't understand it, please reach out to me on Remind so I can give you a quick tutorial. But the amino acid sequence you should have gotten is START, TRP, ASN, ARG, CYS, and then STOP. So remember, on our amino acid chart, we've got start and stop codons. Whenever the ribosome sees AUG, that is the key to start reading that mRNA sequence. Same thing, whenever it sees UGA or another stop codon, that's its signal to stop reading the sequence and move on to the next, right? So keep that in mind as we move forward. If you see a stop codon, nothing afterwards is going to get read. But now let's look at our first mutated sequence. So right now, same thing, pause this video, write down that mRNA sequence, and then highlight the change that you see. All right, so you should have gotten that mRNA sequence is AUG, UAG, AAC, CGC, UGC, and UGA. And I've highlighted the change in red on your screen. So the second base in the second codon has actually been changed. So if we use this mRNA sequence on our amino acid chart, let me move this, see. Wait for it. You should have gotten that the answer is just start and then stop. Right, well, we see that AUG is one of our start codons, right? So AUG tells that ribosome to start reading here, but UGA is one of our stop codons. Or well, it's UAG, I'm sorry. So UAG is a stop codon, which means none of these mRNA sequences are going to be read, and none of those amino acids are going to be brought into this sequence, which means no protein is going to be made. Oh, that's kind of sad. So will there be effects based on this mutation? Yes. The protein wasn't made, which means that is a far cry from what we should have gotten in our original strand. So what type of mutation is this? Well, we saw that one of these bases was substituted or changed, right? Which means that this is a substitution mutation. All right, let's do the same thing one more time. So I've got our original strand up top, and we need to look at our mutated DNA sequence here at the bottom. So same thing, pause this video, fill out the mRNA sequence and the amino acid sequence, and then press play when you're ready to review it. All right, so the mRNA sequence you should have gotten is AUC, CUG, GAA, CCG, CUG, CUG, A. Sounds pretty different from what we got up top, but let's look at our amino acids and see what we should have gotten. All right, so the amino acid sequence you should have gotten is START, LEU, ASP, PRO, and then PRO. Now, this last A right here does not code for any amino acid because it's not a complete codon. All right, so not a codon means no amino acid. Now, we do have some amino acids, but is this amino acid sequence the same as our original one? I see that START is the same, but everything after that is different. Yikes. So will there be effects? Yes, this is a completely different amino acid sequence, which means that different proteins are going to be made. different protein. I'm gonna write yikes. So what type of mutation is this? Well, we see that we actually had an extra base that was added or inserted into our sequence. So what type of mutation is that? Well, that is an insertion mutation. We had an extra base that was inserted into our sequence, but we also need to note that that insertion caused some pretty dramatic effects with the rest of our sequence, right? It actually caused a frame shift mutation. I'm gonna write, well, it's fine in red. Right, so we see that because that one base was inserted, it changed every single amino acid afterwards. There weren't any other similarities. So because of that one single base, it essentially ruined the entire strand. Now, it didn't ruin it. A protein will be made, but definitely not the same protein that we wanted, right? So mutations can have some really, really detrimental effects. But I've got one more example for you. Now, I'm not actually going to run through this one with you. I want you to do this one on your own. Right, so do this on your own. We will go over the answers tomorrow in class, but I want to see if you can figure it out on your own. So same thing we've been doing, go through and figure out the mRNA and amino acid sequences, and then tell me, will there be effects because of this mutation? And what type of mutation is this? So refer to the vocabulary words that we just went over. All right, guys, if you think of any questions you have, please let me know. But other than that, have a wonderful day. Complete your exit ticket, and I will see you guys tomorrow.